Health. Let me do that again. Good morning and thank you for joining us this morning for Winship Grand Rounds. If you're an Emory University or healthcare employee and you would like to receive a CME credit hour for attending today's Grand Round, the login information can be found in the chat feature at the bottom of your screen. If you have any issues with this webinar or the CME login, please send Kadiatu Fufana an email or drop a note via the chat feature. This morning, we are really pleased to welcome Dr. Olorunseun Ogunwobi. Dr. Ogunwobi obtained his master's degree in biomedicine from the University of Hull, United Kingdom. He earned his second master's degree in clinical and translational science from the University of Florida, Gainesville, and a PhD in molecular biology from the University of East Anglia, Norwich, United Kingdom. This is all in addition to obtaining a medical degree from the University of Ibadan, Nigeria, where we met many years ago, more than 30 years ago. He is the founding director of the Untitled College Center for Cancer Health Disparities Research and a tenured professor of biological sciences at Untitled College of the City University of New York. He's also a member of faculty in the biology and biochemistry PhD programs at the Graduate Center of the City University of New York, and an adjunct faculty member in the Department of Medicine at Weill Cornell Medicine. He is a translational cancer biologist whose work focuses on the molecular mechanisms of progression of solid organ cancers with racial disparities and on multiple approaches to leverage non-coding RNA biology for potential clinical applications in cancer. His work has been funded by the NIH, the New York State Carnegie Corporation of New York, and the National Science Foundation, among many others. Dr. Ogunwobi is a contact principal investigator of the Synergistic Partnership for Enhancing Equity in Cancer Health, the speech program, which is funded by a U54 grant, and also the contact PI for many other grants, including a 3U and a co-investigator on multiple R01 grants from the NCI. An author of 68 peer-reviewed journal articles and 2 book chapters, Dr. Ogunwobi has been issued 5 patents, US patents for biotechnology inventions with potential clinical applications in cancer. He is a recipient of several outstanding awards, including the 2022 Hunter College Presidential Award for Excellence in Scholarship and Creative Activity. Please join me in welcoming Dr. Ogunwobi.
Thank you very much, Tunji. That's a really very generous introduction. I'm pleased to be with all of you this morning and to talk about work that my lab has been doing in the area of non-coding RNA biology. This morning, I'll be focusing on 2 projects in which we have opportunities to leverage our understanding for potential clinical applications in cancer. The first part of my talk will focus on what we've been doing in relation to prostate cancer. And my lab is interested in prostate cancer for obvious reasons. It's the most commonly diagnosed cancer in American men, other than skin cancer. But in addition to that, we also know, and I'm sure this audience is particularly aware of the fact that as many as 1 in 9 men will be diagnosed with prostate cancer in their lifetime, and as many as 1 in 41 men will die of prostate cancer. So this is a profound public health problem, but I am particularly interested in this because of the known disparity in prostate cancer. So Black men suffer a 1.7-fold higher prostate cancer incidence than their white counterparts, and a 2.2-fold higher mortality rate than their non-Hispanic white counterparts. This is a profound disparity, and it's actually larger than that's been established for any other common cancer. As to the reasons why this disparity exists, there are many potential reasons that have been uncovered from research. These reasons include unequal access to healthcare, differences in lifestyle risk factors, as well as differences in intrinsic tumor genetics or tumor biology. And that's my particular area of interest. In this regard, my lab has been studying for some time the role of a gene called plasmacytoma variant translocation 1, or PBT1. Now, PBT1 is located at chromosome AQ24, which you can see an illustration of it right here on this slide. Chromosome AQ24 is the most important region of prostate cancer susceptibility in men of African ancestry, and that's a particular reason why we're interested in this chromosomal locus and in the genes that are located there. With PBT1 is one of them. PBT1 is a non-protein coding gene. It's interesting that really at chromosome AQ24, all the other genes are mostly all non-protein coding genes except for CMYC, which is a very well-known oncogene, being the only protein coding gene that's well established at that chromosomal region. The other thing of interest to us, of course, is that PBT1 is upregulated in prostate cancer. And it's also a complex gene, and that complexity attracts my interest. It's got 12 exons and 6 annotated microRNAs. And we and others have shown that they may in fact have different functions. And finally, it's well known to have alternative splicing that takes place at PBT1. Multiple alternatively spliced transcripts are produced from PBT1, and that's of interest, and also relates to some of what I'll be presenting today. But I'd like to start from some preliminary work we did many years ago, in which we actually did an analysis using a panel of prostate epithelial and prostate cancer cell lines to explore the expression profile of all of the exons of PBT1. And we made an interesting observation using this panel of cell lines, where we found very consistently and reproducibly that the only cell line included in our panel that was derived from a black male consistently overexpressed PBT1 exon 9. We're very curious as to why, and wondered if in some way it would help us explain the disparity that we mentioned at the beginning of this talk, and particularly our interest in intrinsic differences in intrinsic tumor biology. And so to further explore this notable finding of significant overexpression of PBT1 exon 9 in the MDA-PCA2B cell line, the only one derived from a black male in our panel, we collaborated with our colleagues at the University of Ibadan, the University College Hospital Ibadan. That's a hospital that serves a predominantly black population in Nigeria, in West Africa. And we were able to obtain normal prostate tissues, benign prostatic hyperplasia tissues, and prostate cancer tissues from them. And then did an analysis of PBT1 exon 9, which we found to be interesting from our analysis of cell lines, and found that in that population, PBT1 exon 9 is actually significantly overexpressed in prostate cancer tissue, suggesting that this observation may in fact be relevant to prostate cancer. Of course, to really establish relevance, we wanted to make sure that this finding is not just an artifact or a coincidental finding, but that it has functional significance. And so what we did with Gage Powell, who had come into the lab as a postdoctoral fellow, was actually have a clone PBT1 into a plasmid vector and then overexpress PBT1 exon 9 in a cell line, the RWP1 cell line, which was the only one in our panel that actually models normal prostate epithelial cells. So this is a non-tumorigenic cell line which models normal prostate epithelial cells. And so Gage was able to overexpress, as we can see in this figure, she was able to overexpress PBT1 exon 9 in this cellular model. And when she did that, the cells became more migratory and they also became more proliferative, which is interesting because increased migration and proliferation are characteristic of prostate carcinogenesis. In addition to this evidence of functionality at a cellular level, we also noticed that overexpression of PBT1 exon 9 induced some molecular changes as well. So, for example, the cells that overexpress PBT1 exon 9 started to express more proliferating cell nuclear antigen, a protein that's a well-known regulator of the cell cycle and may explain why the cells have become more proliferative. We also know that conversely, if you knock down PBT1 exon 9, those cells express less proliferating cell nuclear antigen, which again fits into the fact that PBT1 exon 9 makes the cells more proliferative. But in addition to these functional changes that we see in these bench studies and these molecular changes, we then put the cells into mice. And this is important because RWP1 cells, they're non-tumorigenic prostate epithelial cells. If you put them into mice, we usually use the Norsky gamma mice, they would never form tumors. They're non-tumorigenic. However, when you overexpress PBT1 exon 9, like we did, we found that the mice actually produce tumors from implantation of these RWP1 cells that now overexpress exon 9. In addition to producing tumors, which was very, very notable, an analysis of the tumors showed that the tumors, as you can see in the middle section of this figure, the tumors are invasive. They invade into surrounding smooth muscle, and histopathological analysis reveals, the pathologists tell us that the phenotype is a small cell phenotype, which is also interesting because, as you know, the small cell phenotype is characteristic of neuroendocrine prostate cancer, which is particularly aggressive and is really not responsive to current treatment. So we thought this was interesting, and we pursued this a little bit further, for example, by looking at effects of drug treatments, such as how the cells will respond to second-generation androgen deprivation therapeutics like abiraterone acetate. And what we found is that overexpression of PBT1 exon 9 actually makes the cells resistant to abiraterone acetate, which again adds up with the previous finding of an aggressive phenotype, which is in fact again what we see. Those patients that have the small cell phenotype neuroendocrine prostate cancer, they're generally not responsive to abiraterone acetate, and this is illustrative, we believe, of the role of PBT1 exon 9 in aggressive prostate cancer. We've gone on to look at potential molecular mechanisms for this. We did RNA sequencing analysis in which we found a number of genes are upregulated by overexpression of PBT1 exon 9, and many other genes are downregulated. The notable upregulated genes include CAV14, CAV1, VIM, and FM1, all of which are involved in prostate carcinogenesis. As a notable downregulated gene is Claudin-4, which, as you know, plays a role in cell adhesion, epithelial to mesenchymal transition, and other processes that are involved in cancer metastasis. And so we're chasing this down, these molecular mechanisms. And on the next slide, I summarize some of the things that are going on in the lab in this regard. So, for example, we're interested in determining the molecular mechanisms leading to overexpression of PBT1 exon 9. Which you saw in the analysis of the tissues that we showed you previously. But in addition to that, we are also interested in validating those downstream upregulated and downregulated molecular mechanisms as a result of PBT1 exon 9 overexpression. And actually in figuring out how it is that PBT1 exon 9 downregulates or upregulates those specific genes that we've been able to identify from our RNA sequencing analysis. And the other thing we're currently working on in the lab is determining the functional role of specific PBT1 exon 9 upstream and downstream mechanism, particularly in relation to treatment resistance and metastasis, which we think are key features of clinically significant disease. And then finally, because of our interest in alternative splicing at this gene locus, we're interested in performing 5' or 3' rapid amplification of cDNA ends, the so-called race experiments, to clarify definitively that PBT1 exon 9 may be part of one or more alternatively spliced PBT1 transcripts, and then to determine their relevance functionally and clinically in prostate cancer progression. So these are the interesting aspects of PBT1 exon 9 that we're looking at. But I'd like to just change gears a little bit and talk about another aspect of that gene locus that we came to find interesting from an analysis of the 1,000 of data from the 1,000 Genome Project, where we focused just on the PBT1 locus. We wanted to understand this locus at population level. And we made a very interesting observation in collaborating with Wig and Chu in my department. And the observation we made is that the region of the gene that spans exons 4A and 4B and is painted here in the beige color, we noticed that, first of all, this is a region that's very rich in single nucleotide polymorphism. There's actually a run of about 75 single nucleotide polymorphisms at this region. And then we found that at that region, there's significant genetic differentiation between African populations, and you can see them clustering together here, the red dots at the middle portion of that top part of the figure. But then all the other populations are all clustering together down below. So this was an interesting observation of significant genetic differentiation between African populations on the one hand and all other populations on the other hand. So suggesting some interesting significance perhaps to this region of the PBT1 gene. And so we traced it down a little bit by doing haplotype network analysis with Wig and Chu. And this is what it looks like. Where we were able to, again, do a deep dive into this region that's very rich in single nucleotide polymorphisms. And this haplotype network helped us to identify unique haplotypes. So in fact, we have identified 99 distinct haplotypes at the PBT1 4A4B region. Now, the sizes of the circles that you see on that figure are proportional to haplotype frequencies. So you can see somewhere in the middle there, that's the most frequent haplotype. The high frequency haplotype is somewhat in the middle of the cosmopolitan clade there. And then within the circles, we have enclosed pie charts that show relative frequencies of a haplotype in the 5 population supergroups. And we have a key at the top there that shows the 5 population supergroups that were included in this analysis. And we've also gone on, and I'll show you on the other slide, to actually identify what these haplotypes are that are high frequency haplotypes. And this is the chart that we've been able to pull out. We've named them G3, G30, G12, and so on and so forth. But even more interesting to myself is the fact that we can determine the frequency of these particular haplotypes in the population supergroup. So for example, we know that the G69, the G59, the G62, and the G48 haplotypes are the most enriched in Africans. And the G30, G93, G12 are the least enriched, and they are enriched in the other groups. So what we plan to do with this data is to ask a couple of questions. First of all, do the African enriched PBT1, 4A, 4B haplotypes, such as the G69, do they confer susceptibility to prostate cancer? And conversely, do the haplotypes enriched in the so-called archaic clade, such as the G12, do they confer protection to prostate cancer? And we plan on using a combination of experimental approaches in the lab as well as population type studies to get to the answers to these two questions. In the meantime, we've gone on to utilize the resources we accumulated from our collaboration with our colleagues at the University of Ibadan to continue to look at PBT1 exons 4A and 4B. And so when we look at the tissues, for example, we see specific overexpression of PBT1 exon 4A in a way that actually appears to be prostate cancer specific. You can see that there is significant overexpression if you compare to benign prostatic hyperplasia, and most certainly if you compare to normal prostate tissue for PBT1 exon 4A. And that prostate cancer specific overexpression is also replicated for PBT1 exon 4B. Now, again, is these findings functional? Is there functional significance to this? Well, we leverage the same approach that Gagi had used before, again, in overexpressing PBT1 exons 4A and 4B, using the RWP1 model, the model of non-tumorigenic prostate epithelial cells. She succeeded in overexpressing in that model. And yet again, we found that the cells become more proliferative and they become more migratory. But a notable difference here, whereas PBT1 exon 9 overexpression appear to transform the cells and make them tumorigenic and actually create an aggressive phenotype of prostate cancer in mice, when we implanted these cells overexpressing PBT1 exons 4A and 4B into mice, they were actually not tumorigenic. And so that made us wonder whether in fact they are really non-tumorigenic, or perhaps if they're somewhat less tumorigenic and maybe we needed to allow our experiment to last longer. We're not fully clear about this, and one of the things we like to do in the lab going forward is redesign the animal studies to test the hypothesis that perhaps these are less oncogenic and maybe they need more time or need other factors to be present in the system in order to be oncogenic. So all of this preliminary data actually made us think that we might have an opportunity to address an important question in the field, and that relates to a clinical biomarker that can be used for screening and diagnosis. As we all know, prostate-specific antigen is currently the biomarker that's used. Serum PSA is used currently to help with screening and diagnosis, but it's got significant drawbacks, such as its lack of specificity, which actually results in a high negative biopsy rate. And so there continues to be a big need in the field for a new biomarker assay which can accurately diagnose aggressive prostate cancer and can distinguish. And this is another problem with PSA, is inability to distinguish between disease that will have an indolent cause and one that may turn out to be aggressive and require aggressive treatment. So we wonder if these new biomarkers we're studying may be able to assist in solving this problem. And so to pursue this goal, we thought it would be necessary to create an assay that will likely have translational value or clinical value. Prior to that, as you can see, most of our studies have relied on relative quantification of transcripts using qRT-PCR. But we wanted to develop an assay that can be replicated in labs anyway. And so we came up with this copy number-based quantification assay, in which we leveraged, again, the work that Gagi had done in cloning these transcripts into plasmid constructs that allowed us to be able to have known quantities of these transcripts. And then we used the serial deletion technique to be able to create the standard curves. And this is what a typical standard curve will look like, for example, for analysis of PBT1 exon 4A copy numbers. And using this standard curve, we're able to detect specific copy numbers of PBT1 exon 4A in a variety of sample types, including cell lines, tissues, serum, and plasma. And we pretty much are able to do this for exon 4B and exon 9 as well. And so now with the establishment of this assay, we wanted to check if this assay is indeed useful. We have a good sense for what we find when we do relative quantification using cell lines. So for example, we know that the RWP1 cell line generally expresses less PBT1 exon 4, A4B, or 9 than the MDAPC2B cell line. And so we used our copy number assay to see if we can not only determine copy numbers, but if what we're finding is reflective of what we've known from previous analysis over the years. And indeed, we find that MDAPC2B, which expresses more PBT1 exon 4, it does have more copy numbers of exon 4, and the same is true of exon 4B and exon 9. So that's, really not surprising, but we did that to give us a little more confidence in the assay we had developed. What was interesting, though, is we then collaborated with Emmanuel Asante-Asomani, who is an expert in mathematical modeling, and he applied a methodology called linear decision boundary analysis. And the goal was to use this approach to determine if the copy numbers of PBT1 exon 4A or 4B or 9 that we can determine using this assay, if they would be predictive, and to what extent would they be predictive of whether RWP1 is a non-tumorigenic prostate epithelial cell, which we know it is, or to what extent would they be predictive of another cell line, such as MDAPC2B, that's the cancer cell line. And what we found is that in every case, with 100% certainty, using that linear decision boundary analysis, we can predict accurately which cells are non-tumorigenic and which cells are cancer cells. And this is what the data down below shows, in which Emmanuel Asante-Asomani was always able to use this approach to predict accurately that RWP1 cells are entirely different from the MDAPC2B cells. And so we thought this was interesting because we wondered if this suggests the ability then to use this assay in a predictive way. If serum analysis of the copy numbers of these exons would predict who would have a positive prostate cancer biopsy. And so we collaborated with colleagues at the University of Texas in San Antonio, where they have an early detection research network biorepository. Our colleagues there, Robin Leach and Michael Lees, gave us 40 serum samples. We looked at PBT1 exon 4A copy numbers, and as you can see, by the way, we were blinded to the clinical information of the patients that provided the samples. We just got the serum samples, we did our assay, and then at the completion of the assay, we send the data back to them and they're unblinded. And in this case, there's no real difference in the copy numbers of PBT1 exon 4A that we were able to detect in the serum samples of those who later on turned out to have cancer versus those who did not. However, we do see a significant difference in the copy numbers of PBT1 exon 4B, and it appears that there might be significance also for PBT1 exon 9. Of course, this was an analysis of only 40 samples, and now we've continued our collaboration, and we have expanded this analysis to 200 samples, and we're even looking to do that beyond with an expansion of our collaboration to include colleagues at the MD Anderson Cancer Center and Memorial Sloan Kettering Cancer Center. And now we have the goal of testing the hypothesis that perhaps a composite score that includes copy numbers of PBT1 exon 4A, 4B, and 9 altogether detected in the same serum samples may enable us with some mathematical modeling that Emmanuel will work on to accurately predict prostate cancer and distinguish between indolent and aggressive prostate cancer. Again, with us being particularly interested in being able to develop an assay that will be able to definitively identify patients, Black patients particularly, who may then go on to have aggressive prostate cancer. So this is our goal, And we're continuing to work with the team that you see down there below. In the meantime, we do also have data, and I've just really summarized this because I want to allow some time for questions at the end. We do have some data also looking at the microRNAs that are encoded at this gene locus. We have data on MIR-1205 and MIR-1207-3P. So just a very quick summary of what a graduate student, a PhD student that graduated a number of years ago from the lab, Debash Das did on MIR-1207-3P. And this work has been published. And really the brief summary is that MIR-1207-3P, one of the 6 annotated microRNAs of PVT1 exon 9, is actually underexpressed in prostate cancer tissue. We found that to be the case. But the take-home message from that paper that was published is that this microRNA binds to the 3' untranslated region of the messenger RNA of a gene called fibronectin type 3 domain containing 1. And when it does so, it leads to loss of messenger RNA expression, a loss of protein expression for this gene. And then there is a cascade effect downstream in which there's also loss of expression of fibronectin as well as the androgen receptor. And we believe that this mechanism is very critical for the effect of MIR-1207-3P in inhibiting proliferation, inhibiting migration, and in inducing apoptosis. More recently, though, we've also come to recognize a role for c-MYC downstream of this pathway. And we have a new PhD student in the lab, Priyanka Ghosh, who is working on this, and particularly on the idea that c-MYC, along with the androgen receptor and fibronectin and FNDC1 may in fact be working together as part of a complex. And she does have some interesting data, which I'm unable to present today again because of time. But I would like to show you something, though, an experiment that was done with Debash, in which we had created some synthetic analogs of MIR-1207-3P, as well as nonsense scramble oligonucleotide sequences. And we used this in an animal study in which we implanted C42B cells that model castration-resistant prostate cancer into mice. They developed tumors over 61 days, and then we started to intervene with these oligonucleotides. The MB5 being a synthetic analog of MIR-1207-3P, and MB1 and MB3 being scrambled oligonucleotides. And what you see there is that the mice, all the mice, of course, had tumors, but the mice that received the MB5 intervention, it led to significant inhibition of tumor growth in them. And we also had data that showed that there was inhibition of metastasis in them, and so on and so forth, suggesting that, in fact, MIR-1207-3P may be a tumor suppressive microRNA, which is interesting, given that the PVT1 gene itself is amplified, exons 4A, 4B, and 9 are overexpressed. Now we're seeing MIR-1207-3P being unexpressed and potentially serving as tumor suppression. So we're interested in understanding the mechanisms for those different profiles for various elements at this locus. And again, very quickly, we found MIR-1205 to also likely be a tumor suppressor. And this is data from work that Michelle and I do, who graduated as a PhD student from the lab a number of years ago as well. Again, using synthetic analogs of MIR-1205. In this case, we made a comparison between MB-1205 and a scrambled oligonucleotide that doesn't do anything called MB1, in which we noticed that as soon as we started to intervene in the mice with these oligonucleotides, the mice that received MB-1205, their tumor growth stopped, whereas those that did not continued to have increased tumor growth. Again, this was a model of castration-resistant prostate cancer that we had engrafted into these mice. And so she too pursued an understanding of the downstream molecular mechanism. She actually used an approach in which she used a biotinylator probe of MIR-1205 to pull down, all of the rare molecular targets, and then we used a combination of RNA sequencing as well as specific RT-qPCRs, and we identified that MIR-1205 directly binds to the 3' UTR of a gene, of the messenger RNA of a gene called Fry-like or FREEL. She was actually able to nail this down, not just in the RWP1 model that you see there, but she was able to replicate these findings in the C4-2b model, the PC3 model, as well as the MBA-PCA2b model. So we're pretty confident that MIR-1205 directly binds to FREEL. And that's interesting because we did a collaboration with Andres Bonner, who's at Weill Cornell Medicine. Now, at Weill Cornell Medicine, they actually have a program in which many of their prostate cancer patients from which they can get tissues, maybe from biopsies or resections, they actually do sequencing of these tissues. And what we found from analyzing their data is that neuroendocrine prostate cancer has significant overexpression of FREEL, as does castration-resistant prostate cancer, or FREEL. And this is important because, as we mentioned on the previous slide, MIR-1205, that's encoded at PBT1, directly binds to FREEL. And Michelle, of course, has done a lot of studies in the lab where she was able to demonstrate, and I'll include this in my summary here, that MIR-1205 does play a role in neuroendocrine differentiation of prostate cancer. So in summary, before I go to the concluding part of my talk, in regards to the PBT1 gene locus, I've talked about overexpression of PBT1 exons 4A and 4B in prostate cancer tissue. I've talked about population level differences at that region of the gene and of how overexpression leads of those two exons leads to proliferation and to increased migration. I've also talked about overexpression of PBT1 exon 9 in prostate cancer tissue and how that leads to increased proliferation and migration of prostate epithelial cells and increased expression of proliferation cell nuclear antigen, that's a regulator of the cell cycle, as well as how that likely induces resistance to treatments such as abiraterone acetate, and of course how that ultimately, we believe, leads to transformation of cells and cancer progression, even leading to aggressive prostate cancer phenotype in mice. We've also just very briefly touched on the fact that MIR-1207 is underexpressed in prostate cancer, and that it has a role in inhibiting proliferation, migration, and inducing apoptosis. And I did show you some very brief data of how it's able to suppress prostate cancer growth in vivo, and we believe it does so via this mechanism that Das uncovered and was reported in the paper that I showed you. And then finally, I spoke briefly about MIR-1205 being underexpressed in prostate cancer tissue, how it inhibits castration-resistant prostate cancer growth in vivo, and how it directly binds to the 3' untranslated region of Fry-like. And even though I didn't present the details of the data, we do know that MIR-1205, in fact, loss of its expression induces neuroendocrine differentiation in prostate cancer cells. So I'd just like to very quickly take the last part of my talk to talk about a new project that we've started to work on in the lab. And it relates to our interest in doing genome engineering of the 3' untranslated region of well-established oncogenes with a view to targeting these oncogenes for therapeutic applications in cancer. And the reason why we're interested in this is, as you all know, even in the setting of cancers with known oncogenic drivers for which we've developed molecular targeted therapy, a huge challenge in the field is patients that are resistant to molecular targeted therapy for well-established oncogenes. And a good example of this is ERBB2 or HER2. This is a well-established driver oncogene in a number of cancers, breast, gastric cancer, and so forth. But many patients with ERBB2-driven disease are actually resistant to ERBB2 targeted therapy, such as trastuzumab. And there is also instances in which patients with ERBB2-driven disease actually have low ERBB2 expression, and consequently, their treatment options are limited, even though they have disease that's driven by well-established oncogene for which we have targeted therapy. And because of my interest in cancer disparities, I'm also interested in the fact that there's data now coming out suggesting that there is disparity in response to molecular targeted therapy. For example, in ERBB2-driven breast cancer, where we're noticing that Black women with the same disease are less responsive to molecular targeted therapies, currently available molecular targeted therapies, than their white counterparts. So we believe an approach such as the one we're going to demonstrate here today in the concluding part of this talk may level the playing field and improve treatment outcomes overall for folks that are currently resistant to currently available molecular targeted therapies for ERBB2-driven disease. And the general approach leverages what we already know about the 3' UTR. So, for example, we know that this is a hub for translation, stabilizing, destabilizing, and polyadenylation of transcripts. We already know that there are AU-rich elements at the 3' UTR and that they are involved in nonsense-mediated decay of transcripts. And we know that it does so via a complicated process that involves a variety of enzymes. We also know that this region has motifs that determine which transcripts are stabilized or destabilized and degraded. And we've actually, from the work of Kevin Strulam and others, identified what these specific motifs are that are stabilization motifs, as well as which ones are destabilization motifs. So Chidi, a new postdoc in the lab, has developed this approach in which we've cloned DNA constructs that have these destabilization motifs that will target specific stabilization motifs at the 3' UTR. And we've succeeded in transfecting these constructs into cells. And when we've done so, we've actually, I will show you, successfully degraded the target messenger RNA and the target protein, and consequently led to anti-cancer phenotypes. And the model system that I will be illustrating this data from today is the NCI 81975 cell line, which is a model of non-small cell lung cancer that's driven by ERBB2, also has an EGFR mutation that's targetable ordinarily by osimertinib, except that in this instance is non-responsive to trastuzumab as well as to osimertinib. Really great model of a disease type that we know the driver oncogenes, we have targeted therapies, but the patient is non-responsive to these, or the disease is non-responsive to these. So here's some data from what we've done with this NCI 81975 cell line. And this first piece of data is immunostaining data of ERBB2 and a comparison of cells that in which we transfected these destabilization motifs versus cells that we did not. And we also included a control in which we treated the cells with trastuzumab. So when you treat the cells with trastuzumab, you're unable to affect ERBB2 expression, which shows resistance to trastuzumab. On the other hand, the cells where we transferred these destabilization motifs were successfully able to significantly inhibit ERBB2 protein expression when you compare to the wild type or vector controls. And actually, this data is reliable because when we do Western blotting, which I actually really prefer, we're able to replicate the same data again. Trastuzumab doesn't affect ERBB2 expression, but these destabilization motifs do, and we tried 3 of them successfully. Of course, the direct mechanism for this is via its effect on the messenger RNA, and that's what I showed below, in which, again, the destabilization motifs are able to significantly inhibit transcription of ERBB2 Of course, we don't expect trastuzumab to do that because it's a monoclonal antibody that's supposed to affect the protein, but nevertheless, we included that as a negative control. But what's more interesting is when you do all of this, you actually affect cell viability. With all of the 3 destabilization motifs, we do that. But even beyond affecting cell viability in vitro, we are taking this work into mice because the effects are not just transient, they are stable effects. And so we've implanted these cells into mice, and here's what we find. We find that at the end of the experiment, the mice that received intervention of these destabilization motifs that are cloned into these plasmid vectors, they end up with tumors that are significantly less in volume in comparison to the wild type controls and the control mice that receive the empty vectors. And we plotted that both on the left to show you what happens at the end of the experiment, but also on the right to see what happens all the way through, in which again, the 3 groups of mice that receive these destabilization motifs, tumor growth actually significantly stalled and even appeared to have decreased in one particular group, whereas in the groups, the control groups, tumor growth continued all the way through to the end of the experiment. So this is very promising, we believe, as an approach to inhibit tumor growth in the setting of disease with non-oncogenic drivers, but resistant to current molecular targeted therapy. And we're continuing to do this in a number of other models. In the meantime, we are able to demonstrate that the target is, in fact, we're hitting the target. So this is, so again, we excise the tumors, we stain them for ERB2, and you can see that the tumors from the mice that received the destabilization motifs, ERB2 expressions lost when you compare to the controls, those that didn't receive the intervention or those that just received the empty vector. And this is work again that's been done by Chidi in the lab along with Chika and Barris. We've gone on to actually do RNA sequencing analysis. And what I'm showing here is gene ontology analysis of the RNA sequencing analysis that we did that shows that when we do this approach, there's significant upregulation of genes that are involved in messenger RNA decay, and there is significant downregulation of genes that are related to ERB2 signaling. And you can see something similar when you look at the volcano plot that's on this slide, in which the genes that are significantly downregulated are ERB2. You can see it right at the top there, along with other genes that are related to ERB2 signaling. And then on the right-hand side of the slide, you can see that the significantly upregulated genes, these are all genes that are related to messenger RNA decay. A very good example, and perhaps the most significantly upregulated example, is UPF3B. And if you pull it up using a heat map, down below, you'll see it's like the difference is like between night and day, in which the controls have, the expression is far less. But when you destabilize ERB2, there's significant upregulation of UPF3B. And we've continued on to analyze, for example, because the plasmid vector that was used in this case as a promoter that's driven in part by DCP1A. And we wanted to make sure that DCP1A is not at all involved in the process. And doing simple messenger RNA analysis, we find that we're, of course, able to inhibit messenger RNA expression of ERB2, but DCP1A is not affected at all in the controls or in the samples in which we transfected these destabilization motifs, suggesting to us that the DCP1A in the vector is not a factor in this case. We've then also gone on to do kinase RA analysis because we're interested in the downstream mechanisms that are perturbed by this destabilization of ERB2 messenger RNA expression and protein expression. And also in doing this experiment, we wanted to test whether in fact this approach is specific. we've succeeded in using this approach to target ERBB2, but we've also developed constructs that are successfully able to use a similar approach to target c-MYC. And so in this kinase array analysis, we compared both. And as you can see, the dot plots from the wild type samples as well as samples that just received the empty vector, they're pretty similar. You start to see differences when you look at the dot plots from the samples that were transfected with the destabilization motifs for ERBB2. But the differences you see there are actually different from the differences you see when you target c-MYC. And so on the next slide, if you unblind, we're able to quantify these differences. And of course, there are many of them, but I'd like you to zero in just on Wink1 kinase and Yes kinase. And the reason we're doing this is because Wink1 kinase and Yes kinase are known mechanisms of resistance in patients that have ERBB2-driven disease but are non-responsive to trastuzumab. In this example, we're showing that when we successfully target ERBB2, we're able to, as you can see in the red, we're able to suppress Wink1 kinase expression as well as Yes kinase expression. However, when you target c-MYC, you can't do that. Not only suggesting that we're able to successfully target known mechanisms of resistance, but suggesting that this is in fact specific, which is important, that when you target ERBB2, you're able to successfully downregulate ERBB2 targets. And if you target something else, such as c-MYC, you won't be able to target ERBB2 targets. And we have an ongoing study that actually does tell us the specific c-MYC targets that we're able to target. Because of time, I'm not going to go into that today. However, I'd like to show you on this slide something we think is interesting, and that is the fact that this approach appears to be reprogramming cancer cells to be more like normal cells. So again, the model is the NCH1975 type, which is really a model of non-small cell lung cancer that's driven by ERBB2 and EGFR mutation. And what we've compared with in this case is when we introduced the destabilizer motifs for ERBB2, we've then done a comparison with the BAS2B cell line, which is a model of normal lung epithelial cells. And what you can see here is that the expression profile of ERBB2-related genes looks more in the cells that receive the destabilization motif, now looks more like the expression profile in the BAS2B cell line model. The other thing I'd like to show is some high-resolution genome mapping that we've done. This is an approach called optical genome mapping, and our goal in doing this was to look at genome-wide effects, to look at what this approach does to CMV gains or losses or aneuploidy gains and losses. And we found, interestingly, that this approach appears to improve aneuploidy, which is a problem in cancer. You can see that in comparison to the wild type and vector controls, there is less aneuploidy gain, and there is more aneuploidy loss, and overall, there is less CMV gains and losses, suggesting that, in fact, this overall may have positive genome-wide effects. But of course, we're interested in the specific effects to drill down and validate their positive impact and to rule out any deleterious genome-wide effects. So I'll be concluding at this point and just summarizing this second part of the talk, in which I've talked about our new approach of engineering, stabilizing the ERBB2 triple-nuclear ARE motifs to destabilizing motifs, and to use this approach to decrease ERBB2 transcript and protein expression in ERBB2-driven disease, but that's non-responsive to current molecular targeted therapy. And in the example of the NCI 8-1975 cell line, which is the model of non-small cell lung cancer driven by EB2 and EGFR mutation, we showed that when we did this, we found it led to significant loss of cell viability and inhibition of in vivo tumor growth. And I also showed you that actually downstream of that, there is significant downregulation of EB2-dependent kinases, including YES1 and WINK1, that are known resistance mechanisms in treatment-resistant EB2 expressing cancers. And we believe that upstream, this works via increased expression of messenger RNA decay genes, such as UPF3B. And I showed you finally, by means of optical genome mapping analysis, that we haven't. It appears that at genome-wide level, this is beneficial, and we're interested in further drilling down to really make sure that there are no significant deleterious genome-wide effects. In the meantime, in the lab, we're continuing in vivo studies. I'm particularly interested in exploring multiple delivery mechanisms, such as AAV, and actually we do have some interesting data in a novel approach of packaging this in nano cage. And we found that in the experiment, particular experiment where we did this, we saw enhanced anti-metastatic effects. So we're pursuing this further, and not just with EB2, but we're doing so with CMYC and a number of other well-established oncogenes. So at this point, I'd like to conclude by thanking my lab members, past and present, that have contributed to some of the work that was discussed today, as well as my collaborators nationally and internationally, and the groups that have funded research. Thank you so very much for listening today, and I look forward to some questions.
Fantastic. Thank you very much for that excellent presentation. To those listening, please, if you have any questions, use the Q&A feature, which is located at the bottom of your screen. While we wait on questions, I wanted to remind everyone that next week we have Dr. Karen Jacobson from Harvard University, who will be talking about CAR T cells in the real world, clinical and translational biomarkers of efficacy and toxicity. To view all upcoming Winship Grand Round lectures, please visit the Grand Rounds page on the Winship Cancer Center website or the Winship calendar. While we are waiting for questions, I have one of mine, Sheung. So you truly mentioned the significance, which I think it's going to be very helpful on the population level in differentiating between the indolence and the aggressive prostate cancer phenotypes. Because like you're aware, I mean, a lot of patients will die from something else despite being diagnosed with prostate cancer. But I'm really particularly interested in targeting, especially the microarrays, I think the problem is most of the work in targets, at least genomic targets, have been more successful in oncogenic rather than tumor suppressor genetic aberrations. And so I don't know if either your lab or if you're aware of what others are doing in using tumor suppressor genomes as a target compared to what is currently known.
Yeah, thank you for that question. So, yeah, people always find it easier to target oncogenes because if you identify a tumor suppressor, then you'll be looking to develop approaches to augment the tumor suppressive effect. And in that case, microRNAs are actually attractive because, as I demonstrated, if we identify a microRNA to be a tumor suppressor, what we've demonstrated is that we can develop synthetic analogs of that microRNA that's been demonstrated to be a tumor suppressor. And we've demonstrated both in cell culture and in vivo that if you give those synthetic analogs you're able to demonstrate a tumor suppressive phenotype. Now, the question you would ask is there any data suggesting that this has utility clinically? And the answer is yes. I do not know if any of those have been approved by the FDA, but I'm aware of clinical trials that have demonstrated, I believe in mesothelioma. There was a study that I saw a number of years ago. Yeah, I'm not aware of any of these ended up being FDA approved and in clinical use. But that's the idea that we can develop synthetic analogs of these tumor suppressive microRNAs. And we of course are able to demonstrate efficacy in vitro and in vivo. The question remains as to how efficacious this will be in a clinical setting.
Fantastic. I don't see any questions right now, and I know we're even a few minutes past time. But once again, thank you so much for taking the time to, discuss some of your work. And hopefully there'll be many opportunities for collaboration down the line. And we will be seeing you around. Thanks everyone for taking the time.
Thank you all very much. Have a good day, everybody.